Assalamu alaikum. Today we are going to discuss about chronic inflammation. The basic principle of chronic inflammation is characterized by the presence of lymphocytes and plasma cells in tissue, delayed response but more specific adaptive immunity than acute inflammation. Stimuli include persistent infection, most common cause. Number 2, infection with viruses, mycobacteria, parasites, and fungi. Number 3, autoimmune diseases. Number 4, foreign material. And number 5, some cancers. T lymphocytes produced in bone marrow as progenitor T cells further develop in the thymus where the T cell receptor undergoes rearrangement and progenitor cells become CD4 helper T cells or CD8 cytotoxic cells. T cells use TCR complex, TCR and CD3 for antigen surveillance. TCR complex recognizes antigen presented on MHC molecules. CD4 T cells MHC class 2, CD8 T cells MHC class 1. Activation of T cells require binding of antigen MHC complex and an additional second signal, CD4 helper T cell activation. Extracellular antigen, for example, foreign protein is phagocytosed and presented on MHC class 2 which is expressed by antigen. B7 on APC binds CD28 on CD4 helper T cells providing second activation signal. Activated CD4 helper T cells create cytokines that help inflammation and are divided into two subsets. Number 1, Th1 subset secretes interleukin 2, T cell growth factor and CD8 T cell activator and interferon gamma macrophage activator. Number 2, Th2 subset secretes interleukin 4, facilitates B cell class switching to IgG and IgE. Interleukin 5, eosinophil chemotaxis and activation, maturation of B cells to plasma cells and class switching to IgA. And interleukin 10 inhibits Th1 phenotype. CD8 cytotoxic T cell activation, intracellular antigen derived from proteins in the cytoplasm is processed and presented on MHC class 1 which is expressed by all nucleated cells and platelets. Interleukin 2 from CD4 Th1 cells provides second activation signal. Cytotoxic T cells are activated for killing. Killing occurs through secretion of perforin and granzyme. Perforin creates pores that allow granzyme to enter the target cell, activating apoptosis. Expression of FAS-L, which binds FAS on target cells, activating apoptosis. B lymphocytes. Immature B cells are produced in the bone marrow and undergo immunoglobulin rearrangement to become naive B cells that express surface IgM and IgD. B cell activation occurs through antigen binding by surface IgM or IgD, results in maturation to IgM or IgD secreting plasma cells. B cell antigen presentation to CD4 helper T cells through MHC class 2. CD40 receptor on B cell binds CD40L on helper T cell, providing second activation signal. Helper T cell then secrete interleukin 4 and interleukin 5, mediate B cell isotype switching, hypermutation and maturation to plasma cells. Granulomatous inflammation. Subtype of chronic inflammation characterized by granuloma, which is a collection of epithelioid histiocytes, macrophages with abundant pink cytoplasm, usually surrounded by giant cells and a rim of lymphocytes, divided into non-caseating and caseating subtypes. Non-caseating granulomas lack central necrosis. Common etiologies include reaction to foreign material, sarcoidosis, beryllium exposure, Crohn disease, and cat scratch disease. Caseating granulomas exhibit central necrosis and are characteristic of tuberculosis and fungal infections. Steps involved in granuloma formation: macrophages process and present antigen through MHC class II to CD4 helper T cells. Interaction leads macrophages to secrete interleukin 12, inducing CD4 helper T cells to differentiate into Th1 subtype. Th1 cells secrete interferon gamma, which converts macrophages to epithelioid histiocytes and giant cells.